Good. Well, thank you very much, and thank you for having me. Thoroughly enjoyed the stay. I can't say I enjoyed the trip. Took a little bit longer than expected, but here we are. Yeah, developing an IMTA value chain, sea-based farming of macroalgae and mussels in Saldanha Bay, South Africa. I'm going to keep my talk short and sweet. I think I'm the last presenter today. I don't think there's another presenter after me. Is there another presenter? Okay. There is one more, but it's cancelled. So I am the last speaker of the day, so I'm going to keep it short and sweet because it's been a long conference. So before I kick off, I just want to contextualize the work that I'm going to be talking about this afternoon. I'm a member of the AquaVitae project, which is funded by the European Union. We're 36 partners across 17 different countries. We've got 13 case studies, and they're all working towards developing technology for low-trophic species aquaculture around the Atlantic. And that's exactly where this particular case study fits in. And I'm going to be talking about one of the tasks of this case study. It actually feeds into a couple of other case studies as well, but I'm not going to spend too much time on that. I'm just going to stick to the one task. And I'm taking this opportunity to share some results with you, some of which probably will never be published. So it's a nice opportunity to share information so that it doesn't get lost. We are obviously going to publish our work, but I'm going to first take you through some of the preliminary scoping exercises that we went through, and those are probably never going to be published, and if I don't talk about them, they're never going to be shared. So it's a nice opportunity to share that information, and I'll also share some of the stuff that will later on contribute towards publications that will come out of this work. So before I get into any further detail, I also want to just contextualize aquaculture in South Africa, just so that you know where I'm coming from. Okay, in South Africa, we've got a very, very small aquaculture industry, the biggest of which is the abalone culture industry, where it's a shore-based industry where we pump water into tanks on land. And there's actually, and a little bit earlier today, our sister project Astral mentioned some of the IMTA work that's taking place, large-scale commercial aquaculture that's taking place, IMTA aquaculture in South Africa, and has been taking place for many, many years, and very, very successfully. But that's not what I'm going to be talking about today. The other industries are the bivalves and trout. So I say we're very small. Just to give you an idea, we're producing 2,500 tons of abalone a year. It's very small, but we are the third largest abalone producer in the world. So although it's a small industry, it's niche, it's high value. Mussels is a bit bigger. We're producing about 8,000 tons, but by value, nothing. It doesn't touch the abalone. So what I'm going to be talking about today is taking the mussel industry in South Africa and having a look to see whether it's possible to integrate it or develop technology to make it an integrated system. So firstly, Saldanha Bay you'll find it on the eastern side, on the western side of South Africa. It's pretty much almost the only sheltered bay that we have. So we don't have a coastline that's conducive to shore-based aquaculture. There are a few other much smaller bays. So the culture of mussels is pretty much limited to Saldanha Bay. It's sheltered. It's characterized by upwelling events and very, very nutrient-rich water, so it's highly conducive to mussel farming. At this stage, so there are numerous farms, as I said, about 8,000 tons a year. So I'm not going to spend too much time talking about this. The production cycle, I think, is about 8 months. At the moment, it's monoculture. I'll show you some pictures in a few moments. What makes this one of the more successful aquaculture industries is because of the complete value chain. There's a complete value chain. Everything that you need to farm mussels is there. There's the market, everything. And that's probably why there's been so much failure in other aspects or in other industries in aquaculture in South Africa, but that's another topic because of incomplete value chains. Macroalgae, on the other hand, although it's farmed in tanks, in land-based culture, there's no sea-based farming of macroalgae to the best of my knowledge in South Africa. Certainly not in Saldanha Bay, or I put there not yet, because potentially after this project, we might, if it's successful, we might have algal farming in Saldanha Bay. But the bay is conducive. There's a lot of potential. There are benefits. I'm pretty sure I am talking about IMTA here, even having listened to the definition a few moments ago, because we are utilizing infrastructure. It's a better utilization of resources. It's not your typical high trophic species and low trophic species, but I think we do have two functional groups here. We've got a filter feeder, and we've got algae, which are absorbing dissolved nutrients. And that algae will, in fact, absorb nutrients or metabolites produced by the mussels on these rafts, if the mussels are produced in close proximity. And interestingly, this wasn't an initial motivation for the study, but one of the mussel farmers, and I'm no expert in mussel production myself, but one of the mussel farmers mentioned that there are localized low oxygen conditions occasionally, depending on the sea conditions, in the vicinity of the mussels. And if you've got algae growing right there, possibly that could be another benefit, which isn't a flow of nutrients, but surely that could be included in the benefits of an IMTA system. So it's not, in this case, oxygen as opposed to nutrients. Anyway, I'm getting a little bit sidetracked, so I'm going to get moving again. Our industry partners, Marifeed, are interested in producing Gracilaria because ultimately they want to put it into abalone feeds. There isn't a reliable source other than the Gracilaria that's harvested up off the beaches or imported. So they were kind of interested to see whether it's possible to culture it using these systems. So this was a pilot study. It wasn't replicated. It wasn't controlled. It was simply industry partner going in there, having a look to see whether is it worth actually investing in further research? And I'm just going to show you those results because you'll never see them again probably. Two ropes were basically put into the sea alongside the mussel rafts. They were stocked at two different densities with quite varied conditions between the two. As I said, not replicated. Wasn't intended to be a replicated experiment. So I'm just going to show you some pictures. This is what the ropes look like at seeding. It gives you an idea. On the right-hand side, you'll see one of the rafts with the rope actually lying alongside it. At the time that it was seeded, it was then weighted down, and you can see the Gracilaria. It's a Nephron. Sorry, I forget the name of the rope. It's like a... you can see the Gracilaria being seeded onto that rope, and it then got weighted down. And that's the same rope it had been brought up to the surface a week after seeding it. And you can see that's seeding. That's seeding. That's what it looked like at seeding, and that's what it looked like merely a week later. So we were it looked like quite positive results. 3 and 4 weeks later, very, very clear growth. And there it is at 6 weeks later when they harvested. We worked out we couldn't leave them on these ropes for much longer than 6 weeks because then the Gracilaria actually started to fall off and we started to lose product. So here's a few numbers from that little pilot scoping trial, just to give you an idea of the weight gain in Gracilaria on the two different ropes, quite varied. And also quite varied growth rates. But quite interestingly, and this just describes what went onto those ropes, quite interestingly, it was the rope, obviously, that was stocked with more Gracilaria that grew better. The Gracilaria on that rope grew better. The actual growth rate was better as well. And to be perfectly honest, I'm not 100% sure why. Maybe someone can offer some ideas here. But after that scoping trial, we concluded that Gracilaria was successfully grown. We optimized a few of the production methods to suit the rafting method, the mussel rafting method. One of the constraints we found was in actually drying it afterwards. The mussel, the Gracilaria basically needed to be dried within 3 to 4 hours or it started to rot. So we then scaled that experiment up. We replicated it, this time 26 ropes. I'm not going to go into any details, or else we won't get through the rest of the talk. So pretty much the take-home messages from this preliminary trial was that we could probably produce about 50 to 60 kilograms of dry seaweed per year per raft. The twisted nylon ropes worked way better than the Netlon ropes, the initial ones that I showed you. And while we thought that utilizing the same manpower that was already employed on the mussel rafts would be sufficient, that definitely wasn't the case. It was really quite labor intensive, but possibly could be mechanized and improved if it was ever scaled up. All the other infrastructure was absolutely perfect. The rafts were perfect. The ships and the cranes used for harvesting the mussels could be used for harvesting algae. And so the utilization, the more efficient utilization of space and resources was certainly there. That was one of the main outcomes. We then we started our third sort of loop, or third experiment on a much larger scale this time, more controlled experiments, and everything was nicely replicated. And the next set of experiments, we aimed to optimize the depth at which we lay the ropes. And we also had a look to see whether it would be more efficient to grow the algae on horizontally laid ropes or vertically raised ropes. So this was basically our experimental design, and I spot a mistake there. We actually also stocked at zero at the surface. So we stocked at the surface, 75 centimeters down to 3 meters. And each of those depths was represented by algae that was stocked on, or seeded rather, not stocked fish, on vertical or horizontal ropes. So we ended up with a multifactorial experimental design, and this just shows this is an aerial picture of the and you can you see those are the rafts, and I put a circle around the 3 rafts that we used. So everything was replicated. All the treatments were replicated, and we sort of used a randomized design to ensure that each treatment was represented on at least 3 of the rafts, on each of the 3 rafts. So we, like the other experiments, we harvested Gracilaria from the beach, weighed them out, stocked them, seeded them rather, onto the ropes. This was all standardized. And the next few photos will show the results really nicely. I'll show you some stats in a moment, but I think pictures, sometimes you don't need p-values to see a difference. So after, I think that was a week, so after 3 weeks, you couldn't really see any differences. But have a look at that after 6 weeks. That's the difference in algal growth on the horizontal versus the vertical lines. I mean, it was consistent, replicated across all the rafts. It was the same. So I think that sort of tells a story in itself, and it was backed up. We backed it up with stats, and you can see quite clearly. I think on the left-hand margin, we've just got that's the weight after. That's not 16 weeks, damn it. That was after 3 months. So it's just less than 16 weeks. That's actually 12 weeks. Sorry, that's wrong there. But very clearly, larger weight gain after on the vertical ropes. Again, after 6 months. And interestingly, there was an interaction between the depth and the horizontal and vertical lines. So the red bars on the graph there represent the vertical ropes, and the blue bars represent the vertical ropes. And you can see that on the vertical ropes there was an increase in growth with depth, whereas on the horizontal, consistently, there was actually a decrease, which is quite interesting. So really, the take-home messages at this stage, and this is all very preliminary. The trials are all ongoing. We've started a much bigger commercial scale trial, and we used all of the results from the work that I've just presented with you now. But the take-home method is that the method really has been developed. I think there is potential. It looks like there's potential. The vertical ropes certainly resulted in better production. To date, we've harvested, at the time that this experiment finished, we'd harvested about 220 kilograms of Gracilaria, which has subsequently been put into abalone diets, and that forms part of another case study in the AquaVitae project, where we're feeding this IMTA produced algae to animals both in Spain and in South Africa, which contribute to other case studies. Interestingly, what we didn't realize initially was the current definitely plays a role, and we didn't take that into account. And this picture shows that very first trial that the industry partners did, where they ran a rope down one side. They used the same raft and they ran the rope down the other side. And it so happened that rope 1 was on the receiving side of the current, and rope 2 was on the other side. And didn't pick that up initially. We only picked that up in the second set of trials, and we are now running a way bigger trial, which takes that's upscaled that experiment, and it's taken that multifactorial design and it's taking current and position of the Gracilaria into account. And quite interestingly, if the current runs down the length of the raft, you see a decrease in growth as you go, which I suppose it stands to reason. The other problem that we've come across that we're going to have to address, and that actually might put a stop to this whole idea in the long run, is the natural seeding of mussels, because we're in close proximity to mussels. You've got the ropes lying right next to the mussel beds, and they either grow absolutely brilliantly, the Gracilaria grows brilliantly, or if there's mussel settlement, there's nothing. So that's another problem that we're going to have to address in due course. And that's pretty much me. What we're doing in the current trial, we've scaled everything up. We're doing it on 5 or 6 different rafts at the moment. We're doing a full commercial scale experiment, which sort of represents, I suppose, the third loop of this particular experiment. And it also forms part of a sustainability study where we are measuring the nitrogen, phosphorus, and carbon flow in the system and out of the system. So we're getting quite a lot of really, really good information from this, which we'll hopefully report to you guys in a year's time. Give you some more information. Anyway, thank you very much. That's me.